Both ionotropic and metabotropic receptors can cause a change in membrane potential at the postsynaptic cell. A change in membrane potential caused by synaptic activity is called a postsynaptic potential. Postsynaptic potentials can either be excitatory or inhibitory. An excitatory postsynaptic potential is a depolarization. It drives membrane potential closer to threshold, increasing the probability that the postsynaptic neuron will fire an action potential. The inhibitory postsynaptic potential is a hyperpolarization. It drives membrane potential farther away from threshold, decreasing the probability that the postsynaptic neuron will fire. Whether an EPSP or an IPSP occurs depends on which ions each channel is permeable to. In this case, we see an ionotropic receptor that's permeable to sodium. Sodium will enter the cell because of both diffusion and electrostatic pressure and cause an EPSP. If we have a channel permeable to just potassium, potassium will leave the cell due to diffusion. We know this because the equilibrium potential for potassium is minus 90 millivolts. Positive ion leaving the cell will cause an inhibitory postsynaptic potential, IPSP. If we have an anion channel permeable to chloride, chloride will enter the cell based on diffusion. Negative ion entering the cell will cause a hyperpolarization called an IPSP. Postsynaptic potentials are a subtype of graded potentials. Graded potentials differ from the action potential in that they can vary in size, strength, duration, and polarity. Remember that the action potential is all or nothing, but graded potentials can add together. A small stimulus can cause a small EPSP. A bigger stimulus can cause a bigger or longer EPSP. If the EPSPs add together to reach threshold, then an action potential will fire. If they don't reach threshold, the action potential will not fire. The action potential is a binary yes or no thing. It either happens or doesn't. But EPSPs and IPSPs can vary in size, strength, and duration. Both ionotropic and metabotropic receptors can cause postsynaptic potentials, but these EPSPs look a little bit different. An EPSP caused by an ionotropic receptor tends to be bigger and faster. An EPSP caused by a metabotropic receptor tends to be smaller and slower. And that is simply because of the nature of the receptor. In the case of an ionotropic receptor, it opens when the neurotransmitter binds and closes when the neurotransmitter diffuses away from the binding site. But in the case of a metabotropic receptor, the second messenger that is opened a nearby ion channel could remain bound for longer. Postsynaptic potentials influence the probability that the postsynaptic neuron will reach threshold and fire an action potential. Neural integration is the process of adding together all the postsynaptic potentials to determine whether or not threshold is met at the axon hillock. In this figure from Carlson, we see inhibitory synapses in blue and excitatory synapses in red. And it's trying to show that the charges that are entering as a result of all of these synaptic connections add together. And at the axon hillock, in this case, it says inhibition has counteracted excitation. The action potential is not triggered. Note that we have lots of different synaptic connections on the dendrites, axodendritic. Some of these connections are axodendritic on a dendritic spine, and others are on a smooth dendrite. And we also have axosomatic connections. An interesting thought exercise is to think about which type of synapse can have the biggest impact on whether or not threshold is met at the axon hillock. In this figure from Neuroscience Online, we see the effect of an excitatory neuron in green and an inhibitory neuron in red. When the excitatory neuron fires an action potential, it will release neurotransmitter that will bind to receptors on the postsynaptic neuron and cause a depolarization called an EPSP. If the excitatory neuron fires two action potentials close together in time, it will release more neurotransmitter, which will open more ion channels on the postsynaptic neuron, causing a double EPSP. In this case, adding together both of these two EPSPs, that is temporal summation, reaches threshold, and the postsynaptic neuron fires an action potential. Now in red, when the inhibitory neuron fires an action potential and releases neurotransmitter, that binds to receptors on the postsynaptic neuron, opening ion channels that cause a hyperpolarization called an IPSP. In this example, we see the inhibitory neuron firing and releasing neurotransmitter, and the excitatory neuron firing two action potentials and releasing double neurotransmitter, and the adding together of that one IPSP plus two EPSPs now is a sub-threshold EPSP. We do not reach threshold. We do not fire an action potential. This is a similar figure from another source. And again, we're seeing that EPSPs can add together and IPSPs kind of subtract from that progress. So a lot of communication can be happening between neurons, that is between the green and red presynaptic neurons and the gray postsynaptic neuron, before the postsynaptic neuron actually fires an action potential.